Welcome back to UC Davis Live. We've been hearing a lot recently about new variants of the coronavirus that are more infectious than the original virus. Where do these new variants come from? How concerned should we be about them? What steps can be taken to prevent them from spreading? Well, joining us today to discuss these and other questions are two UC Davis experts in virus genetics and public health. Bart Weimer is professor of population health and reproduction in the UC Davis School of Veterinary Medicine. His research focuses on the population genomics of disease-causing organisms. Since the early days of the pandemic, his lab has been looking at gene sequences from coronaviruses isolated around the world. And returning to UC Davis Live is Dr. Dean Blumberg, professor of pediatrics and chief of pediatric infectious diseases at UC Davis Health. His interests are in preventing and treating childhood infections, vaccine research, and childhood immunization policies. Before we get started, I just wanted to remind viewers that this is a free webcast. You can watch it live and watch it after the live broadcast on the UC Davis Facebook page, Twitter, and YouTube channels. We're aware of others posting links saying that you have to pay to watch this video, so please do not click on those links. This is a free webcast. And we are taking audience questions, so if you're watching us live, just leave them in the comments section and we'll work in as many as we can. Professor Weimer, Dr. Blumberg, thank you so much for joining us today.
Thank you.
Thanks for having me.
So Bart, I'd like to start with you. Could you explain for us what's going on with the virus when it mutates? How does it produce new variants?
Sure. So all viruses, including this one, are very robust in mismatching when their DNA sequence or RNA sequence when they replicate. So this particular virus, it not only is not very good at having fidelity when it replicates, but it also mismatches and mixes up its segments of the genome. So it's very adept at creating mutants. Until genome sequencing came along and really started to be used widely, this was already happening. And so the virus is very good at not being or being able to make its changes very quickly.
Right. So what is it about this virus that allows it to do that more so than other viruses?
Right. So that's a very complicated question. And the short answer is that there's a protein that allows the virus to replicate, and it's not very good at binding its partner RNA. And so it grabs what's around it, makes new versions. And then once those new versions are made, there's small changes known as SNPs or single nucleotide variations. And that's what the variants are that you're seeing today. They're single changes in single bases of this about 25,000 letter genome. So it's very small relative to infectious diseases, but it's very good at changing.
And is this something that we should expect in a pandemic?
Absolutely. So this virus is related to influenza, and influenza, we get a vaccine every year because it does the same thing. So the changes are very often. They change within the outbreak, they change by location, which is why you're seeing the viruses named by the location that they found the variants.
And you've been studying virus strains from around the world during this pandemic. What patterns or trends are you seeing?
So it's already been shown by us and a whole bunch of other people globally that the virus changes about 2 bases per month. So the longer the pandemic goes on, the more changes you're going to see and then will accumulate. So there's not only changes in this genome sequence, but then there's changes in the ability to pass the virus along. So that's what's led to some of the restrictions of social distancing, number of people. So there's 2 components, the genetic changes and the opportunity to then transmit to another person. And so these unique variants will come up both regionally, locally, but they'll also be spread by travel or people moving around as well.
And I'm assuming that the strains or the variants that end up surviving are the ones that are going to be more easily transmissible or possibly more damaging to a person's health.
Yeah, so I think there's a couple things. One is they're more easily transmitted from person to person when given the opportunity, but they're also changing in proteins that interact with your cell when you get infected. So there's been a lot of investigation around the so-called spike protein, which binds to one of the receptors in our cells, and it makes the virus more capable of either staying in our systems longer, invading our cells, a whole host of other things that go on that probably Dean could give you more explanation of the medicine, but from a viral perspective, there's a change in the mutation, and then there's a selection process, a transmission process, and then a selection process.
Right. I just want to remind our audience that we are taking audience questions. So if you're watching us live, just leave them in the comments section and we'll work in as many as we can. So Dean, we just got word yesterday that the South African variant was detected in the San Francisco Bay Area. Previously, it was found in a few other areas in the US. The UK variant is in 34 states, last I saw. There's also the Brazilian variant. Can we just go through each one of those variants, whether they're more contagious and or deadlier than the original virus? So the UK variant, what do we know about it?
So the UK variant has some mutations on it that, as Bart mentioned, it makes it bind more tightly to our ACE2 receptors in our respiratory tract. And it binds more tightly, and then it also fuses with the cell, enters the cell more rapidly. And this results in higher concentrations of the virus. So you can imagine if you're exposed to a higher concentration and it binds more tightly, it's going to be more transmissible. So most of the studies suggest that it's somewhere around 50% more transmissible than previous strains. Initial studies suggested that this UK variant was not associated with more severe disease, but that didn't really make sense because with other infections, if you have a higher concentration of the pathogen, it does result in more severe infection. So the latest information that we have, mostly from the UK, is that it does have 65% increased risk of death.
Wow. Okay, what about the Brazilian variant?
So the Brazilian variant is similar to the South African variant. So when I discuss them, I usually group them together. They are based on the UK variant with an additional mutation. And this additional mutation is in a specific part of the spike protein that appears as if these new variants will escape vaccine-induced immunity or previous infection-induced immunity. So the amount of post-infectious plasma, if you take people who've been immunized or previously infected and you take their plasma, their antibodies, and you see how much it neutralizes these new variants, somewhere between 6 and 9 times less viral neutralization takes place. And we're now getting information from some of the vaccine trials, especially the Oxford AstraZeneca vaccine trial, which took place in South Africa, that they found very little effect against mild or moderate disease, so that they don't think that vaccine is going to work against the South African variant.
That's very concerning. So that means that the companies will need to kind of tweak the vaccine to be a little more tailored to these variants.
Yeah, so as Bart mentioned, a really good analogy is the influenza vaccine. We know that every year we're going to have new strains of the influenza vaccine because that virus is evolving and mutating. And so we update it. So we can easily update these new vaccines. And really one of the advantages that people should realize about these vaccines that are available, especially the messenger RNA vaccines, the Pfizer-BioNTech vaccines and the Moderna vaccines, as well as the Oxford AstraZeneca vaccine, which is not yet available, and the Johnson & Johnson vaccine, not yet available. They're really easy for the vaccine manufacturers to update with the new strain, much easier than influenza vaccines. They can basically do that in a weekend. They can change it. And then, of course, after it's changed, they're going to need to test it and make sure that it's safe and effective, but they can change it very rapidly.
Right. And the difference between, say, the influenza vaccine, which usually researchers have to try to predict which strain is going to be out there. The advantage here is that we kind of already know at least what's happening now, right? So they don't have to try to guess which strain they have to inoculate people against.
Right. So they know which spike protein will be the model for the new vaccines. And then not only that, the manufacturing process, it's just much simpler to scale up with these new technologies compared to influenza vaccines, which take 3 to 6 months really in advance, whereas these can be done very rapidly.
Right. So what effects are we seeing on the pandemic from these new variants? I mean, are the COVID tests people are taking now to see if they are infected, are they as accurate with these variants as they are with the original virus?
Yeah, we're seeing that they're pretty accurate. You know, the UK strain does have a dropout, what's called a S dropout, which is one of the targets of the PCR tests. Doesn't detect that, but that's only particular to one of the PCR assays, and the other PCR assays, for the swabs and the antigen tests, they work very well.
That's really great to hear. So when you're saying that one of them doesn't, that just means that one of the tests doesn't come back, but basically generally the test does come back positive if it is a positive.
So almost all the PCR assays detect more than one target of the virus. And so like you might get a positive for one and a negative for the other. That doesn't really make any sense, right? Because you should get either 2 for 2 or 0 for 2.
Right.
So when that happens, the lab can detect that and figure out why that discrepancy exists.
Okay. Stephen, who's watching on Facebook, is asking, is the South African variant possibly 65% more deadly?
Well, I'm going to be saying this a lot this morning, which is we don't know, which is we do have some more preliminary information from the UK variant, and we have much less severity of illness information from the South African variant and from the Brazilian variant. So we're not sure. There's a theoretical risk that it is more deadly. We just don't have that information.
All right. Bart, how do you think we can adapt our approach to deal with these new variants?
So I think what's happened with COVID that's from a scientific and surveillance perspective very positive. That is that we've jumped into using whole genome sequencing for the variants, and that's the way we're going to detect them. So the UK very early on established a countrywide surveillance program, and they're sequencing thousands of viruses or thousands of samples per week, and they're doing it regionally, and they're I would say probably the model for a newer surveillance system for the future, both for bacterial and viral systems, where when you know what's circulating in the population, then you can very quickly start estimating what might come up. And that's what's happened in the UK. So the UK variant was circulating in the population 2 to 3 months before it started to really rise. And we're publishing a paper right now, and it's very easy to show that it's circulating up and down. You're not quite sure if it's going to rise in the population to the level that it has today, but certainly it has an advantageous mutation, which then with more social contact and transmission has been selected for higher disease rates. So I think the point I'm coming to is that what we see in the future, I think, is going to be a lot more genome sequencing and a lot more terminology of genetic variants. And you shouldn't be scared that they're emerging. What you should be aware of is, are they more dangerous or deadly?
Right. So it almost sounds to me like, we've been, as a nation, we've been building up this infrastructure to deliver the vaccines. We've been building up an infrastructure, I guess, to various degrees in different states to monitor what variants are out there or how prevalent the virus is. Do you have any feeling, either one of you, about improvements to these infrastructures, or are they going in the right direction? Are we where we need to be right now, or is there some room for improvement?
I'll start off. So the CDC started doing whole genome sequencing from a bacterial perspective in 2017 on a statewide basis. So each state has the capability of sequencing lots of samples. There's there's some funding questions around it. Are we going in the right direction? Absolutely. It creates a lot of data to handle. And there takes a lot of coordination to analyze that data. England's done a very good job of that. I think the US is on the right road.
Yeah, and I would just second that. I think, Bart mentioned that the UK was way ahead of us, and that's why they detected that variant, because they looked for variants. If you don't look for it, you can't find it. And so I think the information that we have in the US, it's ramping up, but it's really just, a detection of a variant. It's the tip of the iceberg, because we're not testing a very high proportion of the strain. So if you detect it once, you just have to imagine it's in the population.
Right. So when we hear there are a couple cases in the Bay Area, that, for every case that we detect, there are probably several other cases out there.
Yeah, and I don't know what that means, if there's like 10 times the number of cases, or 100 times, or 1,000, or maybe there's like 10,000 times more the number of cases, but really it's the tip of the iceberg.
Right.
Yeah.
Susan, who's watching on Facebook, asks, so what if you already are fully vaccinated and now these new strains are here? Are you really protected in any way?
Well, I hope we're protected. I'm not 100% sure. You know, we know with the currently available vaccines that they're 94 to 95% effective in protecting against disease. What I'm hoping is even if they're less effective with these new variants, that they'll still provide some partial protection. We know that's true with influenza vaccines, that when the influenza strain, if it moves away from the strain that's included in the vaccine and you get breakthrough illness, if you're vaccinated, you still have milder illness than if you are unvaccinated. So maybe they won't prevent 95% of infections, but maybe they'll prevent 85% of infections. And maybe they won't prevent mild disease, but maybe they'll prevent severe disease and hospitalization. So I think we're going to see some protection. You have to realize these vaccines are based on the whole spike protein. And so when we get these vaccines, we have an immune response to not just one area of the spike protein, but we have an immune response to several areas of the spike protein, whereas a mutation is going to be just one part of the spike protein. So maybe we won't be able to neutralize that part so well, but we're going to have antibodies and immune responses to several other parts of the spike protein. So I would expect at least partial protection.
So let's talk to the people who might be watching saying, oh boy, why should I even get vaccinated if it's going to be changing every month or two? There's going to be a different variant. Can we talk to that person about how important it is still to get vaccinated and to maybe have to follow up with a booster shot or two as we go down the road?
Yeah, 95% protection is pretty darn good. But let's say you only got like 85% or 75% protection. I'd take that. That's pretty darn good too. So even if it's less effective than 95%, it's still really good. And again, we still don't have enough information to know if it's going to provide even more protection against severe disease or hospitalization or death. We just don't have that data yet. And really, isn't that really what it's all about? I wouldn't mind getting COVID if I was guaranteed to get a mild case and just have a cough and sniffles for a few days. That I wouldn't be scared of it if that was the case. But that's not why I'm scared of COVID. I'm scared of COVID because I know all the severe disease that it causes, the cases of pneumonia, the death, the complications. That's what we really need to avoid. So that's worth getting. And the other reason to get it is to decrease transmission. So we're just starting to get some information about how these vaccines result in decreased transmission. The less transmission we have within any community, the less these variants are going to emerge. The only way these variants are going to emerge is if the virus replicates. If it's not being transmitted, it's not going to replicate, and we're not going to have as much pressure to develop these variants.
So are we seeing any kind of definitive data yet about whether or not the vaccine prevents transmission?
We have some data on that. We have some data from the Moderna vaccine study. In that study, people got the first dose of the vaccine, and then at the time of the second dose, 28 days later, they were swabbed. And among those who were asymptomatic and were swabbed, there was 63% reduction in positive swabs in the vaccine group. And again, I think that's just the beginning of the story, because one would expect that it's not just a positive swab that may result in transmission, but being symptomatic would result in a higher rate of transmission. And we don't know the concentration of the virus in those swabs. It's possible that since they had partial immunity, that maybe there was a lower concentration resulting in less transmission. And again, that was after the first dose, not after 2 doses.
Right.
And then the Oxford AstraZeneca vaccine, we do have some transmission data that shows that it probably about 2/3 reduction in transmission with that vaccine. So that's starting to come in. It looks very promising.
Very promising. Barbara from Facebook is asking, do the variants change previous research showing that outdoor distanced interactions are relatively low risk and that surfaces are not a primary route of transmission? For example, touching mail is not very risky.
Yeah, I don't interpret that the variants really change any of that. I mean, the social distancing and the mask wearing should work to prevent transmission with any of the variants. And really, to me, when we hear about these new variants that may be more transmissible, it's just an opportunity to double down on that guidance. And again, I haven't seen anything different about surface transmission. I think at the beginning of this pandemic, everybody was really concerned about any mode of transmission. There were studies that showed viral survival on surfaces, depending on the surface, could be for a day or even longer. But I'm not aware of any outbreaks related to surface transmission. So people should still wash your hands. Don't get me wrong, but that's not the primary mode of transmission. It's via the respiratory route, and that's why the distancing and mask wearing is your best bet to prevent infection, as well as immunization.
Right. Okay. Angela, who's watching on Facebook, is asking, what about people with autoimmune diseases? Will the current vaccines be okay for these people to receive? Or is the Johnson & Johnson one a better bet for people with autoimmune issues?
Yeah, I'm not aware of any different guidance on the basis of vaccine for people with autoimmune disorders. There were a handful of patients in the vaccine studies that had autoimmune disorders, and they were found to react no differently than others. The CDC has guidance saying that those with autoimmune disorders, depending on the disorder and the therapy they're taking, the vaccines might not work as well. If, for example, they're on medication that weakens the immune system, they may not have as strong a response to the vaccine, but they don't expect any increased risk of side effects after immunization, and people with autoimmune disorders may benefit from immunization.
That's good to hear. Mary on Facebook is asking, does the mRNA in the Pfizer and Moderna exactly the same? And if it's not, I guess she's asking if the mRNA in each one of those, the Pfizer and Moderna, are they exactly the same? And if not, is there a place to receive one as a first dose and the other as a second dose to cover more of the spike protein? Also, and then she has a follow-up. So
Yeah, the messenger RNA is slightly different between the two. They were made by different methods, and so they're slightly different. I don't consider the difference as significant. I don't think that mixing and matching will result in an enhanced immune response. That does work with some other vaccines, but they are so similar that I just don't think there's any meaningful difference between the two.
okay, and then she has a follow-up with, regarding COVID vaccines based on more conventional technologies like Novavax or Sinovac, do you think they will offer protection against the variants?
Yeah, well, you just have to do the studies to see, but basically all these vaccines should result in similar immune responses. The differences between the mRNA vaccines, the Moderna, the Pfizer BioNTech vaccine, is that the antigen, the spike protein, is just delivered differently. It's delivered via the messenger RNA, and then our own host cells then manufacture that spike protein, and we have an immune response. So it's very similar to the DNA-based vaccine, the Johnson & Johnson vaccine and the Oxford AstraZeneca. They use a modified cold virus, an adenovirus that can't replicate. That infects our cell. The DNA then gets transcribed for the spike protein, very similar to messenger RNA. The Novavax just purifies that protein and administers that. So for all of them, we're all having an immune response to that spike protein. They really should result in similar responses.
Right. Mary from Facebook again is asking, why are we seeing the UK variant, the South African variant here, travel versus spontaneous mutation? I think what she's trying to say is, why are we seeing these other variants from other parts of the world and we're not seeing variants that are just kind of homegrown, I guess?
We probably are seeing homegrown variants, but because we're not surveilling like the Europeans are, then we're probably not aware of them in our population. That's one. And they probably are occurring and locally. And there are other variants that are occurring absolutely that we're not really talking about because they're circulating, but not necessarily transmission increases or health impact increases. So there's a huge variation in the genome of the viruses now. The UK has done a great job of cataloging all of those changes. And sometimes a variant doesn't necessarily change the protein. So the same protein is produced even though it's from a variant virus or a variant genome. So the variants that Dean's talked about today are really ones that have changed the spike protein to be substantially different in how it interacts with our cells.
And I would just add that, if people want their own variants, we got them, right? We've got a California variant that some
Right.
people may have seen
Made in
in the
USA.
news. Right.
Yeah, we've got more than one California variant, many, many, many. So if you really look at, so the other piece that goes with this is, if you have the genome sequence and you have the variant information, you need what's called the metadata, and you need additional data that goes with it. And that's what's been happening in Europe, but they've been releasing that data on a very consistent basis and allowing the entire scientific community around the globe to then analyze the information and really provide the UK situation with additional information, where the US is not quite to that point yet. We're ramping up our sequencing, but we're not necessarily sharing some of the appropriate information that helps analysis of what's going to happen and where the virus goes and what the transmission rates are going to look like.
So I think Dr. Fauci said yesterday that by April, we should be in a position that anyone who wants to get a vaccine will be able to get a vaccine. So that we're talking at least 6 or 7 weeks out. So do we have any sense of what the chances are that we will have to deal with yet another variant that could be deadlier or more transmissible between now and then?
I'll defer to Bart on that.
I think from a scientific perspective, the emergence of additional variants by that time are likely. What the transmission rates and the health impact will be of those variants, unclear. We just really don't know. But you should expect to see additional variants come up in that amount of time.
Which just emphasizes the fact that we have to all get vaccinated as quickly as possible, basically. Okay.
Absolutely.
Suzanne on Facebook is asking, if I'm vaccinated and my family is not, are they at risk if I return to work in person?
That's been the concern, is that we just don't have enough information on protection from transmission in vaccinated people. And so we do think that people who are vaccinated should continue to follow the social distancing and masking advice. I would say, please go ahead and return to work if you have a safe work environment. I go into work, but I have a very safe work environment. I have a private office. Everybody else in the hallways is always wearing a mask 100% of the time. And I work with medical professionals. These are people who know how to wear a mask, not like sometimes when you'll see, it bothers me so much, a lot of the sporting events where the coaches and others have the mask like below their nose or something like that. That doesn't happen in my environment. Everybody's wearing their mask correctly. So if you're vaccinated, we already know that you have a 95% decreased risk of getting infected. We do believe that there's decreased risk of transmitting to others. And just yesterday, CDC came out with guidance saying that those who are vaccinated, who are fully vaccinated with enough time for the vaccine to kick in, that if they get exposed to somebody who's potentially infectious, they're not recommending that they need to quarantine anymore, because they do believe the protection is sufficient.
That's great. Danny on Facebook is asking, are there any allergy concerns for the vaccines? I heard there were reactions for a person with shellfish allergies in the past.
Yeah, right now, anybody who has any kind of food allergy, we're not concerned. That's not been associated with increased risk of allergic reactions. The main concern is people who've had any kind of significant, serious allergic reaction to any medication, anything that's been injected. And so the difference is, for those, then it's called a precaution to immunization. And what that means is that those who are immunized are still recommended to get immunized. But for most people, we ask them to wait at the vaccination site for 15 minutes, just in case they do start getting an allergic reaction. For those with severe allergies, we ask them to wait at the vaccination site for 30 minutes, an extra 15 minutes because of that. The rate of serious allergic reactions, anaphylactic reactions, is somewhere between 2 and 5 out of every million doses of vaccine. And to put that in perspective, for influenza vaccines, it's about 1 out of every million doses. So it's a little bit higher than that, but it's still a very low rate of serious allergic reactions.
Right. And basically for most people, the overwhelming majority of people, the worst reaction they might get is a little pain at the site of injection, maybe some fever or chills or something like that, just like with a flu vaccine, but that passes within a day or two, right?
Well, that's true for most people with the first dose, but people should be aware that with the second dose, there are more systemic reactions. So people can have things like fatigue, headache, and many people, especially if they're younger, this tends to be more severe, and it may result in you like feeling like you can't go to work the next day. You may be so fatigued. So younger people after the second dose, be prepared for a more severe reaction.
That's interesting. I mean, I would think when I did hear about something about that with the second dose, and I thought, gee, for elderly people, that might be a decision. They might not want to take it because of that concern. But you're saying like older people are not experiencing that on the second dose.
Older people aren't experiencing it as much. Some do, but it's not as frequent to have those serious reactions to the second dose compared to the first dose. It's still better than getting COVID. So I would still
Right.
recommend immunization. And if anybody is like scared of those, COVID is worse than that. And for those who are older and who've been vaccinated with other vaccines, including the current zoster vaccine, Shingrix, most people describe it not as bad as that.
That's good to hear. Michael on Facebook is asking, do you anticipate people will need to be getting annual COVID shots like influenza? And Dr. Blumberg says the vaccines can be tweaked in a weekend, but needs to be tested. How long would it take to produce a tweaked vaccine?
Yeah, so they can tweak the vaccine and make the new vaccine, but then of course you wouldn't want to just manufacture that and distribute millions of doses. You'd want to study it. Just it's a small change, and yet you'd want to make sure that there's still a good immune response and that there's no increased risk of side effects. So you'd have to do the studies. I mean, that would take a couple months at least, I would think. In terms of how long immunity lasts, we just don't know. The CDC's initial recommendations right now, as of yesterday, are that they're counting people to be immune after immunization for just 3 months. And that's based on the information that we already have that if you've had previous COVID infection, that reinfection is extraordinarily rare within the first 90 days after that. So they're treating it similar to that. But we just don't know. We need to do the studies. We need to have longer term follow-up among people who are vaccinated, see what the duration of the immune response. I don't know if getting 2 vaccine doses is going to last 3 months, 6 months, a year, 5 years. The immune responses look to be better than the immune responses following infection. So we would hope for longer term responses.
Right. Let's see. Shugaj9984 from YouTube is asking, while vaccine efficacy is reportedly about 90%, I've read that number needed to treat is anywhere between 119 and 256. I'm not sure which means millions of people. What does this say about vaccine protection afforded to any one individual?
I think for any one individual, we have 95%. You can think of yourself as having 95% decreased risk of infection. So, I can tell you, to me and my coworkers, when we went to the vaccine site in December, when the vaccine was first offered to healthcare workers here at UC Davis Medical Center, that people felt great. You could just see it from the people walking to and from the vaccine site, that they had a little extra bounce in their step. After I received my first vaccine, I felt like I was 10 feet tall, and I'm not. It just made you feel that good, that you could see the light at the end of the tunnel, and that you could feel like now we're getting somewhere. So it feels really good to get immunized. So I would recommend that people do that. Obviously, if you're not exposed, if you're like extreme social distancing and masking all the time, you have decreased risk of infection. But I'd rather do that and also have a 95% decreased risk of infection.
Right. Margaret on Facebook is asking, we've heard of isolated cases of certain species of animals getting COVID-19. Should we be concerned that there will be more incidents of this as the pandemic progresses? Should we be concerned that this could cause more variants to circulate?
So the animals there's coronavirus in animals, no problem. I mean, that's a lot of animals get coronavirus. They're quite different than COVID-19. So you're going to continue to hear about other animals with COVID or with coronavirus. It's yet to be clearly defined what the transmission between the animal population and the human population is going to be. The mink population was one that there's been some demonstrated transmission, especially in Europe. So you should expect to hear some more of that. But coronavirus is widely distributed in the wildlife and the agricultural animal population.
And I just add that in the U.S., all the animals that have been infected, the pets with the SARS-CoV-2, the cause of COVID-19, they've been infected from humans. So it's mostly been cats who've been infected. People are close with their cats. I'm not aware of any pet to human transmission.
I was going to ask you about that, so I'm glad you brought that up. Right? Okay.
Agreed.
So, as we move forward in this, how do you think we are going to progress, say, over the next several months? I mean, it looks like we're heading in the right direction as far as vaccinations, immunizations, tracking variants. How do you think we should be moving forward? Is there anything that we're not doing right now that we should be doing?
I think things are going to get better. We've got 2 vaccines that are available now. We've got 3 other vaccines that we expect to be available over the next couple months. And that's why I think we're talking about April as being a time that we might have enough vaccine capacity for everybody to be able to be immunized who wants a vaccine. We know that even 1 dose of vaccine works very well. We know the newer vaccines that are coming out are easier to store and ship compared to the previous vaccines. The Johnson & Johnson vaccines, just 1 dose instead of 2 doses. That makes things simpler. This is all good news. And the more people that are immunized, the more immunity we'll have within a community. And that means the sooner we'll be able to open things up, that we'll be able to open up businesses, that we'll be able to return to mass gatherings, sporting events, religious services, etc. For children, we already know that that children are not driving this pandemic. We hope to get schools open soon. We think that we can get schools open with very low risk to other students and to teachers and staff, that this can be done. And so this could be good for children and families.
You mentioned the J&J vaccine, the Johnson & Johnson vaccine. So I know when people go to these vaccination sites, they don't have a choice of which vaccine they can get. So for people who are wondering, the Johnson & Johnson one apparently is not quite as, effective as far as protection goes as the others. If someone were to get that vaccine, would they then be able to, say, a few months later, get one of the other vaccines? Is that something that is not contraindicated?
So one of the things to realize is when you see these numbers that these vaccine efficacy studies, they were performed under different conditions. So they're done in different populations. They're done at different time periods where there's different rates of transmission and different pressure for infection. And they have different definitions of efficacy. So they're swabbing with different definitions and doing different surveillance. So they're not directly comparable. So I know it feels good to have a vaccine that is like 95% effective versus one that they say is 77% effective, but you can't directly compare those numbers. And so for all we know, when you do a head-to-head, they might be equivalent. So I would take any available vaccine and then keep up with the information, and this will sort itself out. We'll figure that out later. But for now, I would take any available vaccine. I'd have no problem with that.
That's interesting. I've never heard that about the numbers. So is there any way to kind of crunch the numbers to make them more apples to apples?
Well, you'd have the only way to do that. Well, there's 2 ways to do that. The best way to do that would be to have a head-to-head. So you'd have, half the people getting one vaccine, half the people getting the other vaccine. The people are equivalent. They're at the same time period in similar environments. That would be the way to do that. But obviously, if you do a study, like, for example, in South Africa, and you do another study somewhere else, and the South African strain, variant is escaping vaccine-induced immunity, that vaccine won't look as good. So that's an extreme example of how you can't do that. The other way to do it is you can do a messier study, is once these vaccines are out there and available, the CDC and others can just look, for the whole country and start looking at cases and retrospectively seeing how many cases are occurring with people who've received one vaccine versus another, and get some idea of if one is protecting better than the other.
All right. Okay. Well, Julie on Facebook is asking, do you think the general population will actually be able to get vaccinated this spring/summer? Are there supply issues?
What we're hearing is if we do get approval of these up to 3 additional vaccines, is that there will be adequate supply over the coming months so that people will be able to be vaccinated, get at least their first dose vaccine in the spring.
Right. Okay. Okay. So, any, closing words that you want to let people know as we hear on the news that these variants are coming our way, and they're more contagious, they're more infectious, they're more they cause worse disease. What are your kind of closing thoughts for people to consider?
I would just say that, as Bart has talked about, some of these variants are of no concern whatsoever because they're just minor changes. And so when you hear about a variant in the news, you need to put it in perspective. Why is this one of concern? Is it more transmissible? Is it more deadly? Or is it like just another variant? Is it just like somebody having, brown hair versus black hair? It doesn't really make a difference. So that's one thing to keep in mind with the news. But the other thing is whether we've got the old strains or the new strains that are circulating, social distancing, mask wearing, and vaccination, it's all good.
Right. Bart?
Same comments. I think I would, even though we have a vaccine, I wouldn't get rid of social distancing yet and masking. I stay diligent with that as we already know that variants are circulating in the US. We just may not have found them yet. And so things are coming our way. So keep doing what we're doing, but we should be able to open things up a bit more. We should be able to have friends around. We should be able to be in schools, but don't give up. It's still coming our way.
All right. Well, thank you so much. This has really been great. Thanks so much for coming on today and sharing your work and knowledge.
Thank you.
Thanks a lot.
Bart Weimer is professor of population health and reproduction in the UC Davis School of Veterinary Medicine. His lab has been looking at gene sequences from coronaviruses isolated from around the world. And Dr. Dean Blumberg is professor of pediatrics and chief of pediatric infectious diseases at UC Davis Health. His interests are in preventing and treating childhood infections, vaccine research, and childhood immunization policies. Until next time, I'm Satirius Johnson, and this is UC Davis Live, COVID-19.